Good morning. Today we are discussing translation initiation in prokaryotes. So first coming to the introduction part, what do we mean by translation? Now, translation is a process by which a protein or a polypeptide is synthesized inside a living cell. So the biosynthesis of a protein or a polypeptide inside a living cell is known as translation. The translation is a universal process. Why universal? Because it is taking place in all the living organisms, whether they are prokaryotic or they are eukaryotes, all living organisms, they are undergoing the process of translation. That is, they are synthesizing a protein or a polypeptide, which is carrying out all the metabolic functions of a living organism. Now, what happens in this process, that what happens in translation is that the language of the nucleotide sequence on the messenger RNA is translated into the language of an amino acid sequence. So messenger RNA is acting as a messenger between our genetic material, that is DNA, and the polypeptide chain that is being synthesized. So the information that is contained in our genetic material is first copied into a messenger RNA strand, and then that messenger RNA strand is decoded into an amino acid chain, which is also a polypeptide chain. Now, this process of translation takes place in the cytoplasm where the protein translation machinery is located. Now, as all the biological processes, translation also has three stages. That is initiation, there is elongation, and the third one is termination. So in this particular video, we are talking about the first stage of translation, that is the initiation step. Now, before I move on to the initiation step, there is a step which is known as activation of amino acids. Now, this step is technically not a part of translation, but this is required for translation to proceed. Now, what happens is that in a cell, amino acids are present in an inactive form. So these amino acids, before they are attached to the transfer RNA molecule, they have to be activated. The activation is performed by a specific enzyme. That enzyme is known as amino acyl tRNA synthetase. So for every amino acid in a cell, there is a specific enzyme, that is amino acyl tRNA synthetase. So if we are activating methionine, the enzyme will be methionine tRNA synthetase enzyme. So that enzyme will identify a particular amino acid. It will activate the amino acid, and then it will bind that amino acid to the correct transfer RNA molecule. So for every amino acid, there is a particular enzyme, and for every amino acid, there is a specific transfer RNA present. So once these amino acids are activated by the enzyme, the activated amino acid are then attached to the 3' hydroxyl of the transfer RNA, and this process, this step requires energy, and that energy comes from ATP hydrolysis. Now, once the amino acid is attached to the 3' end of the transfer RNA molecule, we call that transfer RNA molecule as charged. So this diagram shows how the amino acids are activated in the cell. This is an enzyme, that is amino acyl tRNA synthetase, which will be specific for a particular amino acid. This enzyme has got three sites. One is for the attachment of amino acid. Second site is for the attachment of the energy molecule, that is ATP or adenosine triphosphate. And third is for the attachment of the transfer RNA molecule. So in the first step, what is happening is the amino acid attaches to its site. ATP also attaches to the site, and ATP hydrolysis takes place, and pyrophosphate is released. So a molecule of AMP remains attached to the enzyme. The amino acid is now fit into the active site of the enzyme. And the next step is the binding of the transfer RNA molecule. Now, this transfer RNA molecule binds to its particular site on the enzyme. The amino acid, which is now activated, is then transferred to the 3' end of the transfer RNA molecule, and AMP will be released. So we get a charged transfer RNA molecule, which will look like this. This is a transfer RNA molecule. In this image, I've shown the clover leaf model of the transfer RNA molecule. This is the amino acid accepting end of the transfer RNA molecule. Amino acid will be attached to the 3' end of the tRNA molecule with the help of a ester bond. So the carboxy terminus of the amino acid will be attached to the 3' end of the transfer RNA with the help of an ester bond. Now this tRNA molecule, which has an attached amino acid, we call it charged. Now, after the activation is complete, now we move on to the actual translation process. So translation begins with the first step, that is initiation. And initiation will involve all the steps which will be before the formation of a peptide bond between the first 2 amino acids in the peptide chain. So all those steps before the formation of peptide bond are known as initiation steps. So what are the different requirements for initiation? We require a messenger RNA, has the information that has to be decoded into a polypeptide strand. We require the ribosomal subunits. Here because we are talking about prokaryotes, the small ribosomal subunit is 30S and the large ribosomal subunit is 50S. Now, until translation has to take place, these ribosomal subunits remain separated, remain dissociated from each other in the cell, and they will associate only when translation has to take place. That means they will associate only when the 30S subunit has first attached to the messenger RNA and to the initiator tRNA. So there are certain factors which are present. One of these factors is responsible for the separation of the 30S from the 50S subunit till translation has to occur. So we require certain initiation factors for the process of translation. Initiation factors are 3: IF1, IF2, and IF3. Then we require the first transfer RNA molecule that will bind to the initiation codon, which in most of the cases is AUG. And we require energy in the form of a molecule of GTP. So now, what is the role of these 3 initiation factors in the initiation step? There is initiation factor 1. The function of this initiation factor 1 is to block the A site in the ribosome. The ribosome has got 2 sites for the binding of the transfer RNA, for the elongation of the chain. One site is a P site, the other site is the A site. So the initiator tRNA should only bind to the P site. Therefore, what initiation factor 1 does is it blocks the A site in the ribosomes so that only P site is available for the initiator transfer RNA to bind to. The function of initiation factor 2 is to bring in the initiator tRNA to the P site. So it brings the initiator tRNA to the P site, it places it properly on the P site, and for this process, it requires energy, and therefore this comes with a molecule of GTP. Third initiation factor, that is IF3, prevents the binding of the 50S ribosomal subunit to the 30S ribosomal subunit until translation has to take place. That means until the 30th subunit has first bound to the messenger RNA. It has bound to an initiator tRNA. Then only the 50th ribosomal subunit is allowed to bind. So the function of IF3 is to separate the 30th and the 50th subunits till the time the messenger RNA and the initiator tRNA has attached to the small ribosomal subunit. So translation initiation has got three main steps. First is the 30th ribosomal subunit. Along with the initiation factors, it will first find the messenger RNA and attach to the messenger RNA at a particular sequence. Once this is done, the second step is to place the initiator transfer RNA to its correct location. That means to the P site, attached to the initiation codon. Once these two steps are done, the third step is the binding of the large ribosomal subunit, that is the assembly of the entire ribosome and the formation of the 70S initiation complex. So the first step is the binding to the messenger RNA strand. Now 30th ribosomal subunit first attaches to two initiation factors. One is IF1, which will keep on blocking the A site till the P site is occupied by the initiator tRNA. And initiation factor 3 that will prevent the binding of the 50th subunit until it is required. So 30th subunit attaches to two initiation factors, IF1 and IF3. The complex is known as 30th initiation complex. This 30th initiation complex then starts finding the messenger RNA and it binds to the messenger RNA at a particular sequence. And that sequence is known as a Shine-Dalgarno sequence. So the 30th initiation complex does not bind to the start codon. It does not bind to AUG. It binds to a sequence that is present upstream of the start codon. And that sequence in prokaryotes is known as a Shine-Dalgarno sequence. So this is a ribosome binding site for the messenger RNA molecule. Now this sequence is 8 to 12 specific nucleotide sequence which is present upstream of the initiation codon of the messenger RNA molecule. Now complementary to the Shine-Dalgarno sequence is a sequence present in the 16S rRNA, which is a component of the small ribosomal subunit. So the small ribosomal subunit therefore interacts with the Shine-Dalgarno sequence because it has a complementary sequence present in its 16S ribosomal RNA molecule. So this is a 30S subunit shown in this image in yellow. This is a 30S subunit or the small subunit of the ribosome already attached to two initiation factors, IF1 and IF3. And it has a sequence that is UCC, UCCA. Now this sequence is present in the 16S rRNA. And the sequence is complementary to the Shine-Dalgarno sequence, which is also known as a ribosome binding site. So the ribosome binding site is a Shine-Dalgarno sequence, which is AGG, AGGU, having a complementary sequence UCC, UCCA in the 16S rRNA of the 30S ribosomal subunit. And this sequence, as you can see in this picture, it is upstream of the start codon or the initiation codon. So the messenger RNA binds to the small ribosomal subunit at a specific sequence, and that sequence is a Shine-Dalgarno sequence in the transcript. Now, once this step is done, we move on to the second step of initiation, that is a binding of the initiator transfer RNA. The initiation codon, as we know, in most of the cases, is the AUG codon, and the transfer RNA that recognizes, that binds with this codon, is the one that is carrying methionine. So methionine will be the first amino acid which is added, that is amino acid added at the N terminus of the polypeptide chain. So this initiator tRNA will be the tRNA which is charged with methionine. And to distinguish it from other tRNA which are also specific for methionine, this first tRNA molecule has a methionine attached, and that methionine will be formilated. So there is a formyl group added to the methionine, and therefore the tRNA now is charged with a formyl methionine. This tRNA is the initiator tRNA in prokaryotic cells, and that will be taken to the P site. So this image shows the transfer RNA. This is a transfer RNA specific for methionine. The methionine tRNA synthetase enzyme will activate methionine, and it will transfer the activated methionine to the 3' end of this tRNA molecule. And then a formyl group is added to this methionine. So the formyl group is added from 10-formyl tetrahydrofolate. This is a substrate which provides the formyl group. Formyl group is transferred to the methionine, and what is released is tetrahydrofolate, and we get a charged tRNA molecule which has a methionine attached, and that methionine is now formilated. So the initiator tRNA in case of prokaryotes is a formyl methionine transfer RNA molecule. Now this initiator tRNA molecule that is taken to the P site of the ribosome with the help of the initiation factor, that is IF2, and the attachment of the initiator tRNA to the P site requires energy, and that is provided by GTP. So this image shows us the 30S ribosomal subunit still attached to the two initiation factors, attached properly to the messenger RNA at the Shindle-Garneau sequence. And now at the start codon, the initiator tRNA is also attached. It has a bound methionine which is formilated. So initiator tRNA binds to the P site with the help of initiation factor 2 and energy that is provided in the form of GTP hydrolysis. Now the 50S subunit will attach. The 50S ribosomal subunit binds to the 30S mRNA and the initiator tRNA complex using GTP as a source of energy. So GTP is hydrolyzed now. 50S ribosomal subunit attaches to the 30S mRNA and the F-MET tRNA complex. Now, as the 50S subunit attaches, what is released is the initiation factors, that are initiation factor 1 and initiation factor 3. So these initiation factors are released. The 50S subunit, that is a large subunit of the ribosome, it attaches to the small ribosomal subunit. Messenger RNA is placed properly. The first or the initiator tRNA is also placed properly in the P site. And now this complex is known as a 70S initiation complex. So in the 70S initiation complex, what we will have is the P site occupied by the initiator tRNA, 30S ribosomal subunit attached to the 50S ribosomal subunit, and a messenger RNA strand properly placed in between these two ribosomal subunits. So how to summarize the process of initiation of translation in prokaryotes? The first step is we have a 30S ribosomal subunit. It binds to two initiation factors, IF1 and IF3, and then it starts looking for the messenger RNA molecule. It starts finding the messenger RNA and it binds to the messenger RNA at a specific sequence, and that sequence is known as a ribosome binding site, or specifically in prokaryotes, we call it the Shine-Dalgarno sequence. So this sequence has a complementary sequence in the 16S rRNA component of the 30S ribosomal subunit. So once the 30S initiation complex attaches to the messenger RNA, the second step is binding of the initiator tRNA to the start codon, that is AUG. This is brought by initiation factor 2, which brings in a molecule of GTP as well. So this is placed properly on the P site. Now, once this is done, the large ribosomal subunit has to bind. The binding of the large ribosomal subunit requires energy, and that comes from GTP hydrolysis. The factors are released, and we get a initiation complex, which is known as the 70S initiation complex. Now, as the 70S initiation complex is formed, we move on to the second step of translation, that is known as translation elongation, that I will be covering in the next lecture. Thank you.